Yeah, so I would echo about the equity across the globe is important. Access for everybody to treatments, information care is something that we've talked about before. Is getting that, as Dave's alluded to, just getting the right information out that's fair to everybody, so everybody knows what's really happening and what the data means. But the issue, I'd say, my choice for problems that need to be solved would be that if these treatments, now we realise that the earlier somebody can get access to a treatment or the earlier someone is treated, the greater the potential for the treatment to work. So that's the issue of early diagnosis. So if people can get diagnosed as early as possible, and that the issues with that would be to find an effective biomarker that somebody could be diagnosed perhaps pre-symptomatically, and there's some great work going on with that. So that's about raising awareness and continuing with that research to find those markers.
I would say for concern, or it's more of a challenge, I think. We just had the association just had a roundtable on real-world data and registries, and I think what's really apparent is that there are many data collection efforts. Target ALS, David Walk's natural history study, these are mostly United States based, but just giving you an example, ALS TDI, MDA's MOVER study, and now the National Institutes of Health is going to fund a huge natural history study called All ALS. All of them are collecting data, but what's missing is in the field is the analysis of that data. So, are people living longer? How many of all these, between all these 5 that I just talked about, we're talking about, over 3,000, 4,000 people with ALS. How many are on one of the 3 drugs that are approved? 2 of the 3 drugs, or 3 of the 3? And do they have fewer complications if they are on Riluzole and Radicava together? Do they have fewer pneumonia bouts if they're on Relyvrio and Radicava? We don't have these kind of real-world answers. And if we had them, we could then go to regulators and say combination therapies need to be approved, or go to payers and tell insurance companies that combination therapies have to be paid for. So I think the data analysis part is the sort of the lack of data analysis. And I think a solution would be for organizations like ours to provide funding for data analysis. Maybe not fund any more natural history studies, but fund data analysis of the natural history studies that have already collected data.
So I agree with all of the ones that have come before. Is this on? Yeah, it's on. For me, the biggest challenge, I think, is the longevity of funding. It's kind of infrastructure. Because when I employ somebody, they're usually on a fixed-term contract, which may be 18 months or 2 years, and about halfway through that, I then have to try and find funding so that they can carry on. And if I don't do that, all of their knowledge disappears with them when they move, and they may move just because of the insecurity before the contract ends, and then you've not even got the full contract and it's too short to re-employ somebody, etc. What we really need is a proper infrastructure where we know at each major research institution, and even the ones that aren't major, there are staff who are available to do the clinical trials, to do the data analysis, to do the data collection, and that those people are in a secure position. Or they have sufficiently long-term funding that it's not a big issue if they decide to leave early because that's the way their career is going. We need proper infrastructure, and the solution is for the funders to see that as a priority. And that may be difficult for them because they have to answer to their trustees and boards, and they may not realize that actually this long-term funding is what we really need.
One of the challenges I see, I think it leads on from what Kuldip was saying, and it also from Adriano, in that we are collaborating more. There are lots more big collaborative studies happening around the world, but we run the risk of these themselves becoming kind of silos. How do you make the collaborations collaborate as such? So we bring all the data together and make them all compatible as well. All these data sets, sometimes they can't be combined particularly well. So there's no easy fix, obviously, but one way the Alliance is kind of going to start working towards this is we're bringing all the research directors from the funding bodies around the world together on Tuesday for the first time to have a meeting and decide how we can kind of work better together. I mean, I think Kathy calculated we fund something like £100 million worth of research, which is a fair chunk. But it's not just the money, it's the fact that most of the research directors here have their fingers in lots of pies, so they know what's going on and they can help to bring people together. So that, I think, is going to certainly help perhaps to make research around the world come together a bit more. And now I'll pass the same questions on to Alison.
Okay. I'll answer them one at a time. I'm just kidding. What I'm most excited about, honestly, and I felt this at the NEALS conference and definitely here in this room with all of you, is there's an energy. And it's an energy of hope, and it's a shared energy. We have people from 7 countries sitting right here saying we all feel it, and that's contagious. So thank you for doing that. Just the amount of intellect, like you're all geniuses. I'm going to overlook the little microphone things like whose fork is whose. I saw y'all doing over there. There's a fine line. So I'm super excited about that. The challenge is you've all been missing from the conversation. And to back up to a little bit of everything each of you have said, to the start of where Dave started, is that puts people like myself or patient advocates in a position to say, if a trial doesn't meet its endpoints, it doesn't meet its primary, it doesn't meet its secondary endpoint, but we hear a message that says, but it worked for some, and it does show promise, how do we best advocate? Do we advocate for the sponsor to push for another trial, or do we advocate for regulatory flexibility? How do we make decisions? And so, super easy solution. I'm a woman, so I think I know all the answers. Right? Like, it's easy, just do this. But we need you in the conversation. We need scientists to take back the science and help us understand that so that we can make the decisions that we need to make about participating and supporting certain trials, certain organizations, et cetera. So I hope that that's where we're headed, is a mutual trust going forward.
Thank you, Alison. So after hearing from us for quite a while now, we'll now open the floor up to questions. And surprise, surprise,
Marcella.
Marcella is first off.
Oh.
Okay, so usually we always ask you, what can you guys do for us? This time, I want to ask, what can we do for you? What incentives and strategies could we use as members of the Alliance to attract and promote ALS clinical trials in the Global South and in regions where there are not that many clinical trials?
Should I just pick who I want to answer that? Because I'm standing? Okay. Nick, I think you talked about clinical trials, so this might be. Oh, okay.
I'm happy
not
to
picking.
have a go answering
Okay.
that. So I think there's 2 types of clinical trial in broad terms. There's the pharma-sponsored trials, which are commercial, and there's the academic-led trials. And they have slightly different answers to your question. So the pharma-funded ones, we need to have a discussion with pharma about what would make it attractive for them to go to Africa or other places that are underserved for clinical trials. Why aren't they going there at the moment, and what would help them? It may be things like regulatory controls, or it may be other concerns. So addressing those would be important. For academic-led trials, then it's up to the funders to support those trials. For example, I'm currently leading an investigation of an antiretroviral drug for ALS. That's a global study in 3 regions. It doesn't include Asia, it doesn't include Africa. There's no reason why it couldn't, except we haven't got funders from those areas that would fund it. So if the funders could support that with staff and with other support, then we would easily go there. So I think it's, in the end, it's about bodies on the ground and the regulatory. I don't know if Adriano wants to add to that, or Jess.
Yeah, if I can, I have another point that is probably relevant. If you want to have a clinical study which is properly conducted, we need to have very good centers to do that. So a primary problem is to help the centers there to build a good staff in order to be able to conduct the trial. So it's something that is probably an aspect that can be supported by the Alliance, in my view.
Can I just add that if we collectively support things like Janine's been asking for in terms of foundational support now, it may not happen tomorrow, but the more we do those small incremental foundational supports that allow people to build it within, then we will get there faster, I think, as well.
Did you want to add anything, Janine, specifically to that?
No, thanks, David and others. I don't, we don't really have regulatory problems. It's not, well, certainly not in South Africa. And I would think that the big centers that are involved with ALS Africa Network also don't have that. For me, again, I think that David's point is well, would be welcomed in that, having a slightly longer a time frame and just allowing us to investing money in our human capacity growth, so that we can really expand and become fully fledged centres that are trial ready. I mean, in Cape Town, we can do that. We can start a trial tomorrow. We are busy with a biomarker trial. We have been since 2017. But to really expand that to other areas. But even in Cape Town, we have not done any. We have not been involved in any ALS trials, despite the fact that we are involved in other trials such as multiple sclerosis trials, which is really not an African disease, strangely enough. Thank you.
Thank you very much for your discussion. Early diagnosis is very important for ALS. As a clinician, I'm wondering how can we? The diagnostic delay is very important. It seems like a medical problem, but not only medical problem. It's a social impact also. So how can we shorten diagnostic delay? Is Gold Coast criteria works for these things, or do we need new biomarkers? This is my question. Thank you very much.
Yeah, I could start with that. Is that okay?
Yes, yes, yes. Hold it, go.
I think at the recent NEALS conference, somebody showed data that it still takes 12 to 14 months to get diagnosed. It hasn't changed in like 2 decades. And so, and it's both a biology problem, as you said, are there biomarkers, are there, symptoms that we can pick up, and there's a more social problem, right, where depending on how your health system is set up. So, maybe the second one is more difficult to solve. But on the first one, the ALS Association, for example, developed this tool called Think ALS tool. And the idea is that there might be certain symptoms that appear early, and while a person is going through their diagnostic journey in the United States, for example, typically you go to your primary care doctor, they will do a bunch of tests, and then may even send you for scans, then maybe send you to an orthopedic. The orthopedic does tests and scans, may send you through physical therapy. By the time you get to a neurologist, and now the neurologist is doing scans and tests, and that's what the diagnostic delay is coming into. And so we work with experts from around the world to develop this Think ALS tool with the idea that, if we can get the education out, even though it's a rare disease, that, think ALS. And even if you're not sure, refer that person to an ALS clinic. Let the specialist make the diagnosis, instead of you spending months and months on that journey. And so that, that's one way to go after sort of getting the neurologist, general neurologist, informed about that tool, and then looking outwards and going into orthopedic, going into primary care physicians, going into OT, PTs, people who see people with ALS through that journey. I think the social, with the health system, I think those things are going to be there, but we need to do better on the biology side.
Does anybody want to add anything to that?
I'll just shout out the MND Association's Red Flags tool, which is also in Canada we've been able to do one as well. But back on both of your backs, actually, on both the US and the UK. And we've been trying only one slight difference, which is that we've made it a public tool. And what's been great about that is we've actually had some individuals who have reached out to us who have taken the tool from their website, our website. They've started to feel like maybe they have MND ALS. They've gone to their general practitioner and walked through the tool with them and got a referral straight to an ALS specialist clinic. So skipped that whole middle process of being circulating in a system. So we're still trying to learn how to do that. We're getting incredible support from industry who's pushing this out as a tool that was done collaboratively between, I think similar to you, industry, our clinicians, and our organization. But thank you to both of you for setting the tone on that.
No, I was going to go to the next question. We're just going to jump to some online questions, then we'll come back to questions in the room. So, Martina?
Yeah, so we got a few online questions. I'll read the first one. I guess this is for Tim, but anybody can take this. Based on the time to heal for Tufferson realized from Valor, is it fair to say we need to move away from the patient-centric approach of shorter placebo-controlled trials?
I'm happy to take that, unless Adriano wants to, or Janine. So I think the answer is yes. We do need to move away from shorter trials. It's important to have patient-friendly trials, otherwise we won't have people participating in the trials and we'll never find the answer. But equally, it's important to have a trial that can give you the scientific answer to the question you're asking. There's no point just going through the motions for something that won't give you that answer. So part of this is finding that correct balance between how short should a trial be and what other things can we put into the trial to make it patient-friendly, and how long would it be ideally if we were going to try and answer the question scientifically. So for example, people say they don't want to take placebo, which of course they don't. Nobody wants to take a placebo if they've got a condition like ALS. So how can we reduce that? We could increase the chance that you're on the active. You can have 2-to-1 randomization or 3-to-1 randomization. The problem with that is it means you have to recruit more people. And that makes it more expensive and more difficult to do the trial. Another option is, instead of having it 6 months or 9 months, which I still think is probably too short, a year or 18 months and then a crossover, so that, or you go to open label so that everybody has a chance to be on the drug. Again, you need more people if you're doing that because you're diluting the signal you're looking for. So I think there's lots of things we can do, but it's really important that we move away badly enough that my hand is weak. 6 months, it may not have healed completely, and that's just from a single insult of just bruising my nerve. If you've got an ongoing process like ALS, I think 6 months is unrealistic for most drugs. Tofersen is going directly to the target, and we know it affects the target. So, and even that takes 6 months. So that's my view.
Can I ask you a question about that? Since I regretfully have the microphone. So clearly I work with the Healy platform trial, which is iterative 6-month trials. As I hear Merritt and Sabrina talk a lot about, well, we're not proving it works. We want to make sure that we're not wasting time if it doesn't. And so we're weeding out those things that move forward.
Yeah, that does make a difference if you're really asking that question. I still would argue 6 months is probably on the short side for answering that question. The problem isn't so much that. The problem is if a drug company finds that hint of a signal, what does it do with it? What it should do with it is then say, right, we're going to do a formal phase 3 trial. But there's a very strong temptation to just say, well, let's push for licensing on that basis. And that's the worry, because then we may end up with many, many drugs. You know, 5 years' time, maybe we've got 15 drugs that have that kind of level of evidence. And then what do you give people? Because they all have potential side effects. Are we really going to put people on 15 different drugs with weak evidence when it would be better to put them on 2 or 3 drugs that we know actually work? So I think it's important, if we do that, that's made very clear at the beginning that it's not a regulatory trial.
Yeah, so in that case, from what I hear from all of you, that there's agreement in the science community about that the next step after a trial that weeds out what doesn't work and says what potentially could work would be another trial. But how do you get that message? How do we work with the sponsors or the pharma company to understand that?
So we have to ask the pharma companies, but I think the pharma companies know a phase 2 trial, especially a phase 2A trial, is not for licensing because they're not powered to find efficacy. You know, a phase 2B trial can be powered to find efficacy, but again, 6 months, you'd need very, very large numbers to get. When you do a trial, it's a very crude way of saying it, but you need a number of events. But an event, if you're looking at a survival trial, is death. And if you're looking at ALS-FRS as an endpoint, then it's a change in the ALS-FRS-R that you're looking for. If you don't have that level of change, you won't find the effect. So if you're doing that over 6 months, and you need a specific level of events, then you have to have very large numbers to do that for licensing, and we never have that, because that's not the aim of the Healy platform. The aim of the Healy platform is to very quickly give you that answer. Is it worth taking further or not?
Right.
And it's very well designed for that purpose. So we have to make sure it's not abused. And I think we have to speak to pharma and make sure that they understand that.
Thank you for answering that. I hope we're still friends. Jess, we'll go back to you.
Yeah, we got plenty. So another question that came through was, in the UK, what's the delay in the IL-2 study readout?
Okay, it might be the same person asking these questions. So the delay in the IL-2 readout sees that trial, regulatory approval. If the regulators see there's an error or the data collection wasn't properly conducted or there's some queries that aren't answered, they won't license it and you've lost your chance. Now, for the Miracles trial, the funding ran out earlier than it should have, partly because of the pandemic. And that meant that a small academic team have had to take over the role of the CRO because the funding ran out for the CRO. And that small academic team is doing the job of a very large commercial organization. That takes time. If we had the very large organization able to do it, then all of that work would have been done already. I do know the team is working very hard, and they are getting closer to being able to do a submission, either of a publication or the clinical study report, which is what you need for regulatory approval. So that's the delay. It's unfortunate and it's frustrating, but that's the cause of it.
Yep, let's do one more online.
So this is a question, I think it's a good learning opportunity for this topic. Somebody says, I understand that neurofilament light are an issue only in those with SOD1. Patients are asking me if there is a possibility it can also help others.
This is the genius coming out right here.
Well, so, I mean, and this comes back to almost experts ALS is a great example of, you know, if this is, as Dr. Miller explained, that the axons are breaking down and this is the structural component of the neurons is leaking into our biofluids and we can read those and it's at a certain level, depending on your prognosis of your disease or your rate of progression, then we should be able to see a decrease in anyone. And so that's become a a really great surrogate endpoint at this point, meaning it could be something that helps to go alongside clinical evidence. It's not at this point a point where we can say it's a readout of efficacy, and that's why experts is really interesting, because it's exactly what you just said about the Healy trial, which is setting yourself up to say this is where we want to do a really beautiful trial to look for clinical efficacy. But you probably have a better answer.
Yeah, and the neurofilament levels are a speedometer, really. So the faster the progression, the higher the level of neurofilament. That's an important point, I think, to clarify.
And just to really clarify the question, it isn't just for SOD1, it is for everybody with ALS, because it's just damage of the nerves is leading to the neurofilament leaking out.
We can't, just so you know, whoever's standing here, you can't see the audience, so that's why. Oh, we have 20 minutes. Okay, okay. And we did have a question over here that we had slotted for next, and then I'll come back to, right? Oh, you did, okay, perfect.
Yes. Yep.
Hi. Am I on? I am excited about the 2% for genetic ALS. And my question is, can it be possible that in a non-genetic ALS, because of abnormality in the downstream, there could be a production of abnormal associated protein, and how to identify it, and is there a possibility of using 2% in that subgroup of non-genetic ALS?
Did you understand? I just want to repeat to make sure that we all understood that. That she's very excited about tofersen for SOD1, genetic form of ALS, but I believe the question is, can we utilize that for the generic forms of ALS, and where does that take us for all other people with ALS?
Maybe I can answer. Yeah, tofersen is very specific because of the mechanism that has been so well explained by Tim Miller. So it acts on the blocking the pathological protein SOD1. So as far as we know, it's not affecting on subjects that have not these mutations. Anyway, it is possible that future studies will also look at that in order to understand if there is any possibility to use this drug on other patients. But at the moment, there are no indication for patients without SOD1 mutations.
Is that the question, the answer, or were you asking about whether there were things for non-genetic MND in the pipe?
For non-genetic MNDs, there are now several therapies that have been approved in some countries, and other are under study. So probably there will be a lot of, and I think that Amar has said something that is very important, that probably in a few years we are going to have at least 5 to 10 therapies, and there will be problems in terms of the possibility to give them to all patients. But it will be a very good problem for us.
Yeah, and there are. So we mentioned briefly the fuzz, 1% of all MND, SOD1, 2% of all MND, and then the remain a large proportion, over 90%, is related to a protein made from a gene called TDP43, and that's in people who have non-familial MND or a non-known genetic cause, and there are various therapies in the pipeline being worked on that can change or remove the problems from TDP43, which potentially would work in the vast majority of people with ALS MND. We're not there yet at all. That's all being worked on at the moment. There's a really cool piece of work called TDP Reg that's just been published last week or so by Pietro Fratta in the UK, and that's actually what's found in motor neurons is a lowering of TDP protein, and a great team of people, a huge collaborative piece of work, has found a way or a potential therapy that, if it's successful, would work in that it could bring the level of TDP back up, but only in those motor neurons that are affected by the disease. So it's a great way of hitting only the diseased motor neurons. So these are science fiction kind of things that are coming to reality that are going on in labs, as I said before, all over the world. So some amazing people doing incredible things for everybody in the community. So there are things that will come along, we hope, and people working really hard on these non-genetic forms of MND.
The question was also about whether tofersen might be tried, specifically tofersen, or for everyone. Oh, okay, but to see if there's a subset of people who don't have a variation in their SOD1 gene, and see if tofersen will work? SOD1. Yeah. Sure. So there is a body of evidence over many years and a subset of researchers who have shown that there may be abnormal SOD1 in a wide array of individuals, or at least a subgroup of people who don't have SOD1 abnormalities in their genetics. And that is, it's perfect for me to have said this because it's a controversial subject. It's a subject that there's some people who agree with and other people haven't been able to find it. And that's made it difficult to know what to do. I think that Dr. Miller has actually given presentations where he talks about that potential of trying it. I think it would require industry, Biogen, or somebody saying that they want to go forward with that. The question is there enough belief to put someone into a trial to actually do it? Because we have the tool to now try it with tofersen. There is another therapy that is out there in clinical trial right now. It's an antibody, and it's called SOD101. I think that's what it's called. I keep forgetting it right now. And it does recruit people who have quote-unquote sporadic ALS as well, and it's targeting SOD1 as an antibody. So I don't think, I've heard conversations, people talk about it possibly happening at some point, but agree with Dr. Keogh that probably a large swath of the research field is uncertain that there's a true role for SOD1 downstream in propagation of the disease. But we just don't know for sure.
Thank you. Okay, thank you for waiting so patiently.
Thank you. This is Pablo from the ALS Association of Argentina. I'm transmitting an interest from my country. As you well know, South America held very few ALS clinical trials, but we did have a couple. We recently had the pegcetacoplan trial, which unfortunately failed, but it was a good thing to have it. We have the Cardinals ALS trial coming very soon to Argentina. And the first one we had was masitinib, which is a clinical trial that's been going on for a really long time, and it had a couple of interruptions and suspensions, and then it restarted again many times. This is not necessarily something bad, but it's what happened, and it is a very large trial around the world, probably one of the largest we've seen. So I know that you guys have to be very careful in terms of information and stick to the results that have been published, but could you give us some updates on that? Thank you.
Adriano, I hear them saying that's for you.
Yeah, I think that now some studies are coming to South America, and in particular to Argentina. I don't know if in Brazil there are studies. For the trial that was mentioned, Argentina was very important in masitinib in the phase 2 trial. Now a phase 3 trial is ongoing. not going very rapidly because of several problems. But anyway, I think that it's very important to engage countries outside Europe and US and North America really in clinical trials. And as far as I know, the trial in Argentina was conducted very well, and they had a huge number of patients. The only problem was in the phase 2 trial that some patients lived very far away because Argentina is very big, and so there were some dropouts. And this is the reason why we need to have more centers active in big countries so big as Argentina is.
Pablo, just adding to this, for masitinib, it also depends on what the drug is targeting. So for masitinib, it targets this pathway, the intracellular signaling pathway, which is really important for all cells. And so the safety index for a drug like masitinib is really important. If it was just targeting something that was ALS specific, like TDP43 or SOD1, that's different than if you're going after a target that's, that ubiquitous, found in every cell, in every part of the body. You have to be really careful. And so that, that's with masitinib. Not that I think safety is important for every drug. I'm just saying that for masitinib or drugs that are targeting that pathway, understanding the risk-benefit or the safety therapeutic index is going to be really important.
Maybe I can add something about the safety. And I cannot say so much because I am in the data safety monitoring committee of masitinib, and Ben Brooks is the chair. And currently, we did not, at least in ALS, we did not find any reason to stop the trial. So the safety concern is in ALS seems to be not so big as probably is in other diseases. Also because there is probably something related to the dose. In ALS, the dose is lower than in other studies they are doing. But I cannot say nothing more because I have to keep the confidence about that.
Adriano, do you have an idea of the date for a readout from the masitinib trial? Do you know when it might be finished, when we might get some results?
No, it's still ongoing, and there are some problems in recruitment because some countries did not start the trial.
Thank you. Yes, and I heard there was one over here, but Kathy, do you have a question? Oh, Dave's had his hand up. I really can't see you. I'm sorry, Dave. Go ahead.
Hi, David Busak. I'm a patient fellow. Janine, I was really struck by the data that you showed with just the difference in, I'm not sure if it's racial or ancestry. I guess ancestry is the word. The different ancestry groups, as well as the difference in overall levels in Africa perceived or potentially compared to other countries. And I was just curious if there's opportunities to, say, look at the US that also has a lot of people with African ancestry, and looking at data from the US to support Africa, as well as data from Africa to support US and Europe and South America that might have similar kind of ancestry spreads.
Yes, unfortunately, my battery is about to implode on my computer, and the city power is also off. It's supposed to come on at 6, so I might just suddenly drop off. So I'm sorry. There's limited information on African Americans, but it seems to be certainly it is. Well, I'm sure that the North Americans on the panel can talk to that as well. But the incidence appears to be slightly lower in the African Americans amongst European Americans. I'm not aware of their age at onset, but it is definitely a definite biological signal amongst our African ancestry people that is seen in different parts of Africa. So it's not just in one part. And what is also interesting is that amongst Europeans, the younger onset people usually have a higher chance of having an ALS gene, and amongst our patients, the younger people do not have an ALS gene. So the missing heritability is higher amongst those people with young onset ALS, which is even more intriguing.
Thank you. I think we have...
Thanks for the question.
Yes, thank you. I think we're going to try to take one more online question. I'm sorry if we don't get to everybody, but these guys will be here all week. You can catch up with them and ask your questions. We'll take one more online.
So here's another question. We know that 10% of patients have mutations. We know that mutations also occur in sporadic patients. Which mechanism works in the remaining patients with ALS, and how can we move forward in treatment without knowing this? Good luck.
One moment.
Okay.
Anyone?
So it's actually about 20% of people have a genetic basis, not 10%, because although if you have a family history, then you're much more likely to have a proper genetic basis that we can identify. You can also have a genetic basis without a family history because just having a gene isn't enough to cause the disease. You need other factors. So the question as to what's the mechanism in the people who don't have a genetic basis, I think was answered by David earlier with the TDP-reg issue. So, was it you, Nick? Sorry. Sorry. So TDP-43 is a protein that should normally be in the nucleus of the cell and should then regulate how the genes are switched on and off. And in about 98% of people with ALS, it's not in the nucleus, it moves out into the outer part of the cell called the cytoplasm, and it doesn't work properly as a result. And we don't know what the downstream aspect of that is, definitely, but it looks like at least one of the important outcomes of that is that the genes aren't switched on and off properly, and they have bits of them that are activated that are normally silent, and that makes the proteins work differently. And so what Pietro Fratta's work has done is identified that mechanism as a kind of general problem in ALS, and so if we can target that, we may well have a way of treating all ALS. So I think it's very hopeful. How do you get to that? But at least we're now understanding much more about the mechanism of ALS. So I think we are getting much, much closer to the target and knowing how to tackle it.
I'm sorry, we're going to take one more because I was just notified that, Kathy, you've had your hand up the whole time, and we don't want to do that to you. So if you could talk fast and you could answer fast, go. Okay. Yes.
I asked this question in Orlando 8 years ago at the same session, so I'm going to try it again. Okay. You talk about patient centricity, and I appreciate all the work you do, and I know that you're thinking about patients all the time. But if you look at all of the natural history studies and all the observational studies we have, people like David and Sunny put their data in this one, and then for this one, and then for this one. That's not patient-centric. What are the prospects, especially with the new honking big natural history study that we're getting, and I'm glad we're getting, that we can have an on-ramp so that don't wait until you fill your silos to collaborate. Let's get some collaboration on the front end so that they put their data in once. We have one entity doing the hard work of getting their medical records stuff together and have an on-ramp, and I think that gives you better uptake on all your studies. So thank you, and please don't make me come back in 8 years.
Thank you. I think that's what I just learned this word. It's a BHAG, right? But
Nadia, do you want to jump in on this one?
Nadia?
Okay. Well, I think my first thought was, yeah, we have bigger natural history studies, and some of that actually answers the previous question on, how do we figure out what's going on with the rest of ALS, right? And so these studies are critical to solving the 80% that may not have a known genetic component at this point. Ultimately, there's probably a bigger genetic component, and there may be genes that aren't as largely involved. But anyway, yes, we need to get more people involved in these studies. I think one of the things to consider is that we might actually need several natural history studies because they don't all measure the same thing. And I think that's hard to understand. So when you're contributing data sets to different studies, they might be for different measures. And so I think that's one thing that's important. Another thing is that we need multiple studies because there are times when if we want to validate the findings from a single study, then we need to look to a different data set to validate that, right? And so it might be helpful to have more than one study. At the same time, we need to find a way to not make it so redundant and to get everybody involved in some kind of research. So I think there's a couple of ways to think about that question.
And we really need to make sure those studies can talk to each other, that the data can be combined, which sometimes can be a problem, certainly with patient registries, that it's really difficult to combine the data. So I think we have to think when we build new studies that the data, the questions we're asking or the data we're collecting is going to be useful to everyone else as well. I think that it can be easily used across different studies.
And can I just add to that, which is that Orla Hardiman in Ireland is leading a project which we're all meeting about, I think, tomorrow, called Precision ALS, which is trying to do exactly that. So it's bringing together all the natural history studies of Europe into one study.
Wonderful. Yep. One entry point.
If I can I would add something. That's the project that has been mentioned by Amari is very important, and there is a problem which is related to the different GDPR between US and Europe. So it's becoming very difficult to exchange data across the ocean and probably to other countries, in particular because in Europe GDPR is very, very strict. So this is a point that could become a major challenge in the future. And I think again that some pressure by the ALS associations in everywhere in the world will be very helpful to modify and to attenuate these rules that can really block research.
Yeah, thank you. And just to clarify that, I think, Kathy, what you're saying is the entry point should be centralized, not just the bring it all together afterwards. So that's a good problem to work on going forward. I know we have big plans tonight, and I've been given it's time to go. So thank you all for participating. I'm going to turn it over to Gethin to just close us out.
Great, thank you. And that was a fantastic conversation. I think we probably could have gone on for another 4 hours or something, but obviously there's other things to do tonight. So I'd just like to draw your attention to, in January, we're having a clinical trials update webinar, so you can even hear some more about the clinical trials hanging around the world. That's in January. And there will be more SAC updates coming as well, and more briefing notes coming out with information about the trials. So I think we'll close it off there and just give a quick bit of information again about the dinner tonight. So for PALs, they'll be picked up at 5:30 outside the door. There'll be some 5:15. Sorry, 5:15. Which is 5 minutes. So there'll be 3 small buses and some smaller vehicles for those of you who want to walk. There'll be shuttles afterwards as well, starting from 8:15, and the last shuttle leaving at 9:30 to get you back to your hotels. For those walking to the dinner, Michael from ALS Schweiz will be waiting in the lobby at 5:40, is it? In a red coat, apparently. So look for the man in a red coat. And if you can, be there by 6, because there's going to be a surprise, which it sounds exciting. Could be Santa. You never know. Man in a red coat and a red hat. Okay, thank you all for your attention. I know it's been the end of a long day, but I thought that was a fantastic session. And thank you to all our panelists for fantastic contributions.
Thank you.